Hi class, this is your instructor, Scholar Huff, and we have now made it to the 36th chapter, and this is Roots and Mineral Nutrition. As we begin, please pay close attention to the key concepts here. Namely, root structure is related to function. The primary tissues, epidermis, cortex, endodermis, pericycle, xylem, and phloem of roots, they develop from root apical meristems, and those secondary tissues, the wood and bark of woody roots, develop from those lateral meristems. What an amazing review that was. So as I do continue on, roots associate and interact with many organisms, including soil bacteria and fungi, and I'll go ahead and tell you now, those are some of the most amazing things you'll learn about in the course, and you'll hear that you've heard some of this before. And lastly, soil, the layer of the Earth's crust, has been modified by contact with weather, wind, water, and organisms. So, of course, they contain most of the essential elements required by plants. Let us begin. Roots. So that they're branching underground root systems, and they are often more extensive than the plant's aerial parts. In other words, you might see a plant that is an approximate 4 to 5 inches above the ground. However, its roots, they may extend as far as 8, 9, or even, of course, 1 foot beneath the ground. They may. Roots of a corn plant, for example, may grow to a depth of about 8 feet and spread outward up to 4 feet from the stem. I can hardly wait for the corn to mature. It's not quite there yet, but it is about a foot off the ground now. It's pretty tall. So it states thereafter, the desert-dwelling tamarisk trees reportedly have roots that grow to a depth of 163 feet to tap underground water. That is so deep. 163 feet. That's deep, 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 deep. So the extent of a plant's root depth and spread varies considerably among different species and even among different individuals in the same species. Because roots are usually underground and out of sight, people do not always appreciate the important functions they perform. First, as anyone who has ever pulled weeds can attest, roots anchor a plant securely in the soil. I know when I pull weeds, I wait for it to rain, or at least after I've watered the grass. Otherwise, those roots will keep that plant anchored in, and you'll only get the top part of the plant, and of course the plant will remain there. So a plant needs a solid foundation from which to grow. Firm anchorage is essential to a plant's survival so that the stem remains upright, enabling leaves to absorb sunlight effectively. Second, roots absorb water and dissolve minerals, those inorganic nutrients such as nitrates, phosphates, and sulfates, which are necessary for synthesizing important organic molecules. These dissolved minerals are then transported throughout the plant in the xylem. Many roots perform the function of storage. Carrots, sweet potatoes especially, cassava, and other root crops are important sources of human food. Gotta have food. As I say it that way, if you're looking at the cassava now. So those are the cassava roots. So these plants have been dug up to show the growth habit of the storage roots. Cassava roots are an important staple in the tropics. Some cassava roots contain a poison called prussic acid, which is destroyed during cooking. Surplus sugars produced in the leaves by photosynthesis are transported in the phloem to the roots for food storage, usually as starch or sucrose, until needed. Other plants, particularly those living in arid regions, possess storage roots adapted to store water. In certain species, roots are modified for their functions than anchorage. It may be a function such as absorption, conduction, and storage. So to reiterate, those roots that function, they function for other things than simply storage, conduction, absorption, and anchorage. Those roots may be specialized to perform uncommon functions, which is about where we'll end the chapter. So let's now get to root structure and function. So 2 types of root systems exist. They are the taproot system and the fibrous root system. As I've mentioned these, please bear in mind that we are preparing for your final exam. So the taproot system, it has one main root that formed from the seedling's embryonic root, the radicle. Lateral roots branch out, and this is characteristic of the dicots and gymnosperms. For example, what we have here is the dandelion, the hickory. Most young trees have a taproot and those very large lateral roots. The fibrous root system. It has several roots developing from the stem with lateral roots. So just moments ago, you were shown what is called the taproot system here at A. We're now at 36.1b, looking at the fibrous root system. So, in saying such, it's short-lived embryonic roots, and this is characteristic of monocots. So they're called adventitious roots. They develop from the stem, and they were not pre-existing roots at all. For example, this is found in crabgrass, which of course you may hate, or just not like that much, and have to cut. And even in onions, which I do indeed love. I've planted well over 100 onions in the bag. So taproots have adapted to obtain water deep underground, where fibrous roots are relatively close to the surface, obtaining rainwater. So having put it that way, it's these roots that have evolved, adapted to obtain water in those differing sections of the soil. Roots have root caps and root hairs. The root tips are covered by a root cap. This is review, which protects the delicate root apical meristem. As I've said it that way, please ensure that you all have seen figure 36.2a and b, which I'm going there here shortly. There we are. Having said that, the root hairs increase the absorptive capacity of roots by increasing their surface area in contact with the moist soil. So it's because of the need to adapt to the soil environment instead of the atmospheric environment that roots have those several structures, such as the root caps and hairs, that shoots lack. Although stems and leaves have various types of hairs, they are distinct from the root hairs in both structure and function. So as the root grows, pushing its way through the soil, the cells of the root cap are sloughed off by the frictional resistance of the soil particles and are replaced by new cells formed by the root apical meristem. The root cap cells secrete lubricating polysaccharides that reduce friction as the root passes through the soil. So it's that root cap which also appears to be involved in orienting the root so that it grows downward. And how amazing it is that every root that's a root grows in the downward fashion, or the downward direction. So when a root cap is removed, the root apical meristem grows a new cap. However, until the root cap has regenerated, the root grows randomly, rather than in the direction of gravity. So to continue on where I left off with what are called the root hairs, those hairs continually form in the area of cell maturation closest to the root tip. So this is to replace those that are dying off at the more mature end of the root hair zone. Each root hair is short, typically less than 1 centimeter, or about 1/4 of an inch in length. But they are quite numerous. And when I say they're quite numerous, root hairs greatly increase the absorptive capacity of roots by increasing their surface area in contact with the moist soil, as I just stated, to reiterate that. So the soil particles are coated with microscopically thin layer of water in which minerals are dissolved. So the root hairs establish that intimate contact with soil particles, which allows for efficient absorption of water and minerals. They also modify the composition of the soil by exuding a diverse array of organic molecules. So if you are interested in studying such things, the model for this is the angiosperm, Arabidopsis thaliana. It has been learned that the spatial arrangement of root hairs on the root epidermis is a consequence of complex interactions between cells that form root hairs and the adjacent cells that do not. many genes are involved that code for those transcription factors. So nextly, I'll get to that vascular tissue arrangement that distinguishes roots of herbaceous plants, those being roots from monocots and those of dicots. So what I'll say is that most herbaceous, I repeat, and before I go there, these are the root hairs. I almost forgot to mention this photograph. I was so stuck on the other picture. But as you do look closely here, there is that epidermis with those cells with their cell walls. And then coming from those are the root hairs indeed, increasing the absorptive capacity of roots with soil air, water, and other particles within the soil. Back to where I left off. So I was meaning that most herbaceous dicot roots, they have that central core of the vascular tissue that lacks a pith. So what I'm getting to now is going to be what you all will see for lab, meaning as you all complete lab, this is what it will be. So do indeed learn this, and if you have questions, please ask. So I'll begin with what is called the dicot root. So what we're looking at here is a cross-section of ranunculus. So this is figure 36.3, figure 36.3, A and B. Textbook page 746. So as you go through this with me, I'd recommend you draw it. However, I can't make you draw it, but if you are attempting to earn a high score in the course, drawing is ensuing. Let us continue. So I'll first begin with drawing that epidermis. Draw the epidermis of this called ranunculus. That's the genus. It's the buttercup. So as you've drawn it, that outermost portion is the epidermis indeed. So just inside the epidermis, all of the portion you see highlighted here is what makes up the cortex. This will be that cortex of this herbaceous dicot root. And then just inside the cortex is what's called the stele. S-T-E-L-E. So that is the cross-section of a dicot root. So from here, I'm going to zoom myself in to the stele of the dicot root. And this is zoomed in quite far. This is at an approximate 25 micrometers. How close we are. And to make sure I do my due diligence and review, figure 36.3a, keep in mind that the cortex is what encompasses or what composes the bulk of this herbaceous dicot root. So I say to help you all with this, please make a note somewhere and write it big. The X is your key. The X is what keys you in on is this the root of a dicot or is this the root of a monocot? So that X-shaped xylem in the center lets you know right there and then this has to be a dicot. Someone once said that X marks a spot. So from here, I'll move on down to figure 36.3b, the stele. So here in the stele, I'll begin with the cortex, which we just mentioned. The cortex lines that outermost portion. It's just outside the stele. And now I'll get inside just a bit. So first things first, I'll begin with the endodermis. So it's that endodermis that makes up that inner portion, meaning what's just inside the cortex that is right around that X-shaped xylem. So I'll get right to it. So this X, it has those vessel elements. That's what these cells are. These are cells of the vessel elements that make up the xylem. And I'll say it again, it's the X that lets you know, is this a monocot or dicot root? Continuing on, just inside the endodermis is what is called the pericycle. So we have the endodermis, which is here, and just inside the endodermis is what is known as the pericycle. And then what flanks the X would be called the phloem. So those are phloem cells you're seeing that flank in the invaginations of this X. There you have it. This is the dicot root cross section. For those taking notes, add to your notes now, please. So I've already mentioned that the majority of the dicot root is cortex. I'll continue myself now, saying that dicot stele. So with the pericycle that's there, the pericycle is that outermost layer of the stele. I mentioned that moments ago. And is made mostly of parenchyma cells, which give rise to secondary growth as well as lateral roots. You all know about the xylem and phloem, so I'll go on over to what is the endodermis, just inside. So the endodermis is what regulates the movement of water and minerals. I'll move my cell phone over. So with that, if you have questions, I say please ask. And one thing that I will mention is the key point. Key point here on page 765. It states that the symplast pathway consists of living cells that are interconnected by plasmodesmata. And the apoplasts, that pathway, is what uses cell walls and intercellular spaces. So I mentioned these because the primary function of the root cortex is starch storage. So if you look closely, you're seeing both those pathways. You're seeing both that symplast pathway as well as the apoplast pathway. It is that symplast pathway that is in the blue, using plasmodesmata. The symplast pathway that is in the blue. And it is that apoplast pathway that is in red, using those intercellular spaces of the cell wall. So this is an important picture. And I say this is an important picture because it's integrally important for learning where we're going. So with that, I'll say just one more time that since this is a herbaceous, I repeat, herbaceous dicot root, yes, it has that single layer, the epidermis that's there. It covers the roots. The root hairs are that modification to root epidermis that enables it to absorb more water from the soil. And it is the root epidermis that does not secrete a thick, waxy cuticle in the region of the root hairs, because this layer would impede the absorption of water from the soil. So it lacks that cuticle, and it has that presence of hairs to increase absorption. So if we begin here at those root hairs, as you see the root hair in this photograph being shown here, this is figure 36.4. Beginning at that root hair, most of the water enters the root and moves along the cell walls rather than entering the cells themselves. So a major component of the cell wall is cellulose. Please go back to Biology 103 class. A major component there is. So this is what absorbs water, as does a sponge. It's amazing how cellulose does it. An example of the absorptive properties of cellulose is found in cotton balls, which is almost pure cellulose. I say again, if you've ever had a cotton ball, it's almost pure cellulose. Thanks a lot, cotton, which of course is the fruit. I'll continue. So the root cortex is made up of primarily loosely packed parenchyma cells. So they compose the bulk of the herbaceous dicot root. And the roots usually lack supporting parenchyma cells, and it's probably because the soil supports the root. Although roots, as they age, may develop some sclerenchyma, something for support, the primary function of the root cortex is storage. Storage. And the storage of what? Starch. So starch is that insoluble carbohydrate composed of glucose subunits and is that most common form of stored energy in plants. Yet again, another throwback to Biology 103, you all. So when used at a later time, these reserves provide energy for such activities as growth and cell replacement following injury. Amazing these things are called roots. So I mentioned already the symplast pathway, which is that continuum of living cytoplasm, which is connected from one cell to the next by those cytoplasmic bridges called plasmodesmata. Some dissolved mineral ions move from the epidermis through the cortex via the symplast pathway. The apoplast consists of the interconnected porous cell walls of a plant along which water and mineral ions flow freely. The water and mineral ions can diffuse across the cortex without ever crossing a plasma membrane to enter a living cell. These make up the entire plant body. So I'll now get on into what is called the endodermis. So as I now mentioned that endodermis, it's that inner layer of the cortex, and it is the endodermis that regulates, as I mentioned earlier, the movement of water and minerals that enter the xylem in the root's center. So the endodermal cells, they fit snugly against one another, and each has a band-like region called the Casparian strip. Yes, indeed. So on its radial side and the transverse, which are the upper and lower walls, the Casparian strips, they contain suberin, the waterproofer I mentioned way ago. So it's that fatty material, and I mentioned again, that is waterproof. So until the endodermis is reached, most of the water and dissolved minerals have to travel along the apoplast, therefore have not passed through a plasma membrane or entered the cytoplasm of a root cell. However, the waterproof Casparian strip on the radial and transverse walls of the endodermal cells prevents water and minerals from continuing to move passively along the cell walls. For substances to move farther into the root interior, they must move from the cell walls into the cytoplasm of the endodermal cells. So water enters by osmosis, both directly across the lipid bilayer of the endodermal plasma membrane and through aquaporins. Those are the endocrine membrane proteins that facilitate the rapid transport of water across the membrane. It reminds me of human anatomy and physiology with aquaporins in the urinary system. As in water movement across the lipid bilayer, water moves through aquaporins only along the osmotic gradient from a region of, here we go again, higher water concentration to a region of lower water concentration. Boy, that is an amazing review there. So the key role of aquaporins in the uptake of water by root cells is currently under active study. So at the center of the dicot root is the stele, or the vascular cylinder. So it's a central cylinder of vascular tissues. So the outermost layer of the stele is the pericycle, which is just inside the endodermis. So the pericycle consists of that single layer of parenchyma cells I mentioned earlier, you all, that give rise to lateral roots, also called branch roots. So those lateral roots originate when cells in a portion of the pericycle start dividing. So as it grows, the lateral roots push through several layers of the root tissue, the endodermis, yes indeed, the cortex, and ultimately the what? Yes, the epidermis, before entering the soil. So each lateral root has all of the structures and features: the root cap, root hairs, epidermis, cortex, endodermis, and what else? The pericycle with xylem and phloem of the larger root from which it emerges. In addition to producing lateral roots, the pericycle is involved in forming the lateral meristems that produce secondary growth in woody roots. So there it is for you, class. Please take care of yourselves and study. I'll continue on now. So after passing through I guess I'll go to the endodermal cells. Water enters the root xylem. This is helping you on your test where you will trace the path of a drop of water through the plant. I'll say it again. After passing through the endodermal cells, water enters the root xylem, often at one of the xylem arms. Up to this point, the path of water has been horizontal from the soil into the center of the root. Well, now we'll get more specific. So to help you all out, class, with the path of water, I'll repeat, to help you all out with the path of water, please add this now from our prior chapter. I paused to allow you to get there. What you should add would be, of course, this is water that enters in from the soil. It firstly touches what are called the root hairs, or the epidermis. I say again, it passes through the root hairs, or the epidermis. From there, it enters into what is called the cortex. I will look for this on your test. Thereafter, it enters into what is known as the endodermis. It enters into what is known as the endodermis. From there, after entering into the endodermis, it enters into what is known as the pericycle. And from the pericycle, it then enters into what is known as the root xylem. So from here, the water will go through and go to the rest of the plant. So you can pick that portion up with where we were in the last chapter, being the 35th chapter of the textbook. Stem structure and transport. Well, all right. So from here, we'll move on into, I guess the last thing I'll mention that I almost, I guess I'll say went without, is that once water enters into the xylem, it is transported upward through the root xylem to the stem xylem and from here to the rest of the plant. Class, we did that in the 35th chapter. So one direction of phloem conduction is from the leaves, where the sugar is made by way of photosynthesis, to the root, where sugar is used for growth and the maintenance of root tissues, or stored, usually as starch. Another direction of phloem conduction is from the root itself, where sugar is stored as starch, to other parts of the plant, where sugar is used for growth and maintenance of tissues. So I mentioned this to get to the vascular cambium, which gives rise to secondary tissues in woody plants. It is sandwiched between the xylem and phloem. This is why we labeled it in lab. So because it has an inner core of vascular tissue, the primary dicot root lacks a pith, that ground tissue that is found in the center of many stems and roots. So from here, keep in mind that monocot roots, they vary considerably in internal structure compared to the dicot roots. The layers found in some monocot roots, starting from the outside, are the epidermis, the cortex, endodermis, and pericycle. Unlike the xylem in herbaceous dicot roots, the xylem in many monocot roots does not form a solid cylinder in the center. Instead, the phloem and xylem are in separate alternating bundles arranged around a central pith, which contains parenchyma cells. Because virtually no monocots have secondary growth, no vascular cambium exists in the monocot root, despite their lack of secondary growth. Long-lived monocots, such as the palms, yes, palm trees are monocots, they may have thickened roots produced by a modified form of primary growth in which parenchyma cells in the cortex divide and enlarge. So as we are here, I will further now get into what is known as the monocot root. So here we are. I'll begin at the outside, and if you don't have your pencil and pen ready, please press pause. I'll begin at the epidermis. So we have the epidermis that is there. And if you ever wonder, why is he doing this? Because I know it's on your lecture test as well as your lab test. That is class epidermis. Just inside the epidermis is where we get to the cortex. So the cortex that's there, it's just as you saw ago with that dicot root, how large it is. And then after the cortex, we get into what is the endodermis, that outer portion. And then just inside the endodermis is the pericycle. So since I'm going outside in, I'll now begin at the phloem. I did say it. I'll begin at the phloem, outside in, the phloem. So the phloem is here, and then just inside the phloem is the xylem. The xylem vessel element. And then we end there at the very center with the pith, P-I-T-C-H, the pith. So that is what is inside the stele of a monocot root. Again, the cortex is, of course, extended in size. So from here, I'll continue on. Woody plants have roots with secondary growth. Plants that produce stems with secondary growth also produce roots with secondary growth. These plants that do such things are known as the gymnosperms and woody dicots. They have primary growth at apical meristems and secondary growth at lateral meristems. So the production of secondary tissues occurs at some distance back from the root tips and results from the activity of the same two lateral meristems found in woody stems. It is the vascular cambium and cork cambium. I hope you know those for your test. So major roots of trees are often massive and have both wood and bark. In temperate climates, the wood of both roots and stem exhibits annual rings in cross-section. Before secondary growth starts in a root, the vascular cambium is sandwiched between the primary xylem and primary phloem. As I say this, please see Figure 36.9. So, at the onset of secondary growth, the vascular cambium extends out to the pericycle, which develops into the vascular cambium opposite the xylem arms. As this occurs, the pericycle links the separate sections of vascular cambium, so the vascular cambium becomes a continuous, non-circular loop of cells in cross-section. So as that vascular cambium divides to produce secondary tissues, it eventually forms that thin, continuous cylinder of vascular cambium that continues to divide, producing secondary xylem, called the wood, to the inside, and the secondary phloem, called the inner bark, to the outside. The root increases in girth, and the vascular cambium continues to move outward. Up next, the epidermis, the cortex, the endodermis, and primary phloem are gradually torn apart as the root increases in girth. So the root epidermis is replaced by the periderm, composed of cork cells and cork parenchyma. This is straight from the last chapter, being the chapter called chapter 33. And a bit of this was presented in chapter 35. So both of which are produced by the cork cambium. So the cork cambium in the root initially arises from regions of, or at least regions in the pericycle. So there we are. So we'll next get to some roots are specialized for unusual functions. Adventitious roots often arise from the nodes of stems. Many aerial adventitious roots are adapted for functions other than anchorage, absorption, conduction, or storage. From our foreshadowing to begin the chapter. Prop roots are adventitious roots that develop from branches or the vertical stem, and they grow downward into the soil to help support the plant in an upright position. Prop roots are more common in monocots than dicots. So we find prop roots in corn and sorghum, both of which are monocots and are herbaceous plants that produce prop roots. And its many tropical and subtropical dicot trees, such as the red mangrove, which also produces the prop roots. And that is what you all see here in figure 3610A. Excuse me, that's a screw pine. That is not a mangrove. That is a screw pine. That is a screw pine. 3610A. The roots of many tropical rainforest trees are shallow and concentrated near the surface in a mat, only a few centimeters or so thick. The root mat catches and absorbs almost all minerals released from leaves by decomposition. Swollen bases or braces called buttress roots hold up the trees. They hold them upright and aid in the extensive distribution of shallow roots. So we have prop roots and buttress roots. We're now at the swampy and tidal environments. So in swampy and or tidal environments where the soil is flooded or waterlogged, some roots grow upward until they are above the high tide level. Even though roots live in the soil, they still require oxygen for aerobic respiration. Flooded soils are depleted of oxygen, so these aerial breathing roots, known as pneumatophores, may assist with getting oxygen. So these aerial breathing roots, known as pneumatophores, do such. So they assist those submerged roots. The pneumatophores, which also help anchor the plant, have a well-developed system of internal air spaces that is continuous with the submerged parts of the root, presumably allowing gas exchange. So the black mangroves and white mangroves are examples with pneumatophores. Please see figure 3610C. Up next, climbing plants and epiphytes. If you have not heard of an epiphytic plant, epiphytic plants are those plants that grow attached to other plants, and they have aerial roots that anchor the plant to the bark, branch, or other surface on which they are growing. Some epiphytes have aerial roots specialized for functions other than anchorage. Certain epiphytic orchids, for example, have photosynthetic roots. Those epiphytic roots may also absorb moisture as well. Some parasitic epiphytes, such as the mistletoe, which I know you all have seen, please walk outside, look up at an oak tree, you will see the mistletoe there. They have modified roots that penetrate the host plant tissues and absorb water. Another plant that starts its life as an epiphyte is the strangler fig, which produces long roots that eventually reach the ground and anchor the plant. Now a tree rather than an epiphyte in the soil. So the tree on which the strangler fig originally grew is often killed as the strangler fig grows around it, competing with it for light and other resources. How amazing it is. So we're moving on now to root associations and interactions. So as the roots of individual plants extend to the soil, they often encounter the roots of related or unrelated plants. The nature of these interactions is largely unknown. However, there are several studies which suggest that roots are involved in complex ways, sometimes recognizing the identity of neighboring roots or modifying their growth accordingly. In this example, the wild strawberry roots have been demonstrated to grow more vigorously when they encounter ground ivy roots. In contrast, root growth of ground ivy is inhibited by strawberry roots. Such root associations affect the community dynamics in natural ecosystems and may affect the productivity of agricultural systems. The roots of terrestrial plants species may form mutualistic, meaning mutually beneficial relationships, you all. So this is with certain soil microorganisms. One of two is the mycorrhizal fungi. You all should remember the mycorrhizae. You very well should remember this from the 29th chapter. And of course the Rhizobial Bacteria, which you all should also remember just a bit of from earlier chapters. So these mutualistic relationships provide plants with access to the essential minerals that are limited in supply in the soil. Let us begin. So we'll begin here with the mycorrhizae, which facilitate the uptake of essential minerals by roots. The subterranean associations between roots and certain fungi, known as mycorrhizae, are found in virtually all, I repeat all, I'll say one more time, all terrestrial ecosystems. Mycorrhizae permit the transfer of materials such as carbon compounds produced by photosynthesis from roots to the fungus. At the same time, the essential minerals, such as phosphorus, move from the fungus to the roots of the host plant. The thread-like body of the fungal partner extends into the soil, extracting those minerals well beyond the reach of the plant's roots. In some mycorrhizae, the fungal mycelium, yes, I just said the mycelium, encircles the root like a sheath. In others, the fungus penetrates the cell walls of the root cortex and forms branching arbuscles. Hence, they were known as the arbuscular fungi. So as I say that, I just have to show you here the arbuscles. And you see the arbuscles there. The mycorrhizal relationships are ancient. Some of the earliest known plant fossils, about 400 million years old, contain mycorrhizal fungi in the roots. The mycorrhizal relationship is mutually beneficial because when mycorrhizae are not present, neither the fungus or the plant grows well. The hyphal network of the mycorrhizae appears to interconnect the roots of different plant species in the community so that carbon compounds may flow from one plant to another through their mutual fungal partner. It is so amazing how these work together. And I'll mention that it's in the genome of an ectomycorrhizal fungi. Lectoria bicolor has been sequenced, and scientists have identified many genes that code for small secreted proteins that may be involved in establishing the mutualistic relationships between the fungus and its plant partner. For example, it has been learned that one such protein is secreted by the fungus in response to signals that diffuse from plant roots. So the fungal protein is then uptaken by the plant root cells by endocytosis and enters the nuclei, where it affects the transcription of plant genes that promote the mutualistic mycorrhizal relationship. How amazing it is. Let's get the second of two. So the second of two will be how Rhizobial bacteria fix nitrogen in the roots of leguminous plants. Certain nitrogen-fixing bacteria, collectively called rhizobia, form associations with the roots of leguminous plants such as soybeans, peas, clover, and the list goes on. Nodules are those swellings that house millions of the rhizobia as they develop on the roots. Evidence indicates that the rhizobial relationship evolved much later than the mycorrhizal relationship. So unlike the mycorrhizae, which infect most plant species, rhizobia almost exclusively infect plants of the legume family, which evolved about 70 million years ago. A few other plant bacterial associations that fix nitrogen have also evolved. As with mycorrhizae, the association between the nitrogen-fixing bacteria and the roots of legumes is mutually beneficial. So here we go. Please prepare yourselves for notes. Bacteria receive photosynthetic products from plants, while helping plants meet their nitrogen requirements by producing ammonia in the form of NH3 from atmospheric nitrogen. It is so amazing. So you should remember this from our biogeochemical cycle, lecture. For several decades, biologists have been studying the molecular basis of associations between plants and nitrogen-fixing bacteria. The two partners in this association use cell signaling, that is, a molecular dialogue back and forth, to establish contact and develop those nodules. So pay close attention, class, to what is called figure 3612. Let us begin. So when the soil contains a low level of nitrogen, legume roots secrete a chemical, and the chemical attractants are called flavonoids, toward which the rhizobial bacteria swim. I did just say that. So as that occurs, we then continue moving. The initial signal molecules produced by the rhizobial bacteria are called nodulation factors. So those nodulation factors, or nod factors, they help those root hair cells of the leguminous plants recognize the presence of the bacteria. So recognition is the first step in the infection, I said in the infection by rhizobia, and the development of root nodules. The nod factor apparently attaches to a plant receptor on the root hair plasma membrane, which you all can now see in number 3. This attachment triggers a calcium-dependent signal transduction cascade within root cells that alters gene expression and metabolism, resulting in nodule formation. And please look closely as you see the developing nodule at number 6. The plant hormone cytokinin, which triggers cell division, is involved in root nodule formation. Some genes are involved in the initiation of both mycorrhizal and rhizobial associations. And I say because both mycorrhizae and rhizobial infect plant roots, some biologists have hypothesized that genes and signal transduction components have involved in mycorrhizal associations may have been recruited for the rhizobial relationships that evolved much later. So there is experimental evidence to support this hypothesis. And some of the same genes that trigger infection and changes in root development to form rhizobial nodules are also involved in establishing mycorrhizal infections. So evidence includes several legume mutants that biologists have identified, and these mutants lack the ability to develop nodules for rhizobial bacteria and cannot form mycorrhizal associations. So from here, the soil environment. I'll do the soil environment rather briefly and quickly. So now that we're in the soil environment, the place in which roots live, or at least most roots, the soil is a relatively thin layer of the Earth's crust, and it has been modified by the natural actions of weather, wind, water, and organisms. So it's easy to take soil for granted. So we walk on it, over it, and throughout our lives, but rarely do we stop and think about how important it is to our survival and the vast number of kinds of organisms that colonize the soil and depend on it for shelter and food. Most plants anchor themselves in the soil and from it they receive water and minerals. Almost all of the elements essential for plant growth are obtained directly from the soil, with few exceptions. Plants cannot survive on their own without soil, and because we depend on plants for our food, neither could humans exist without soil. So to be brief, organisms produce carbon dioxide, which diffuses into the soil and reacts with soil water to form carbonic acid. So the lichens produce other acids, and those acids etch tiny cracks into the rock surface, and then water seeps into the cracks, and then the small plants send roots into the cracks, enlarging those cracks. So the topography also contributes to soil formation, meaning soil and rock that's transported down slopes by gravity, and even water runoff from precipitation tends to increase soil erosion on steep slopes, and the moderate slopes may encourage soil formation. So it is soil that forms constantly and change as the weathering of parent material beneath the soil adds to new soil. It kind of reminds me of evolution. The things that shaped the past class are still shaping things today. So there it is. We have evaporation and transpiration with precipitation, plant litter, and of course earthworms with runoff. All of these things contributing, of course, to soil formation. So the composition of soil, i.e. what is soil? I did not say dirt. Soil includes about 25% air and 25% water. Thereafter, inorganic mineral particles, inorganic mineral particles make up about 45% of that typical soil, and only about 5% of soil is organic matter. So I would hope that when you plant your plants, you don't just go out there and say, I need some dirt. You need more than dirt. You need soil. So those minerals are continually cycled from the soil to the organisms, which use them in those biological processes. How amazing it is. So the plants, animals, fungi, and microorganisms that inhabit the soil and interact with it, and those minerals are being cycled, which use their biological processes. So when the animals die, the bacteria and other soil organisms decompose the remains, returning those minerals to the soil. The circle of life, they say. So because different rocks are made of different minerals, soils vary in mineral composition and chemical properties. So with that, the texture of a soil is determined by the percentages of sand, silt, and clay. And I would love to do a lab on sand, silt, and clay, content of your soil at the homes that you indeed live in. So it alone contains an approximate 40% of each sand and silt, and then 20% clay. This takes me back to grad school doing sand, silt, clay separations on my plots. So generally, the larger particles provide structural support, aeration, and permeability to the soil, whereas the smaller particles bind to aggregates or clumps and hold minerals and water. So soil with larger proportions of sand are not desirable for most plants because they do not hold water and mineral ions well. Plants grown in such soils are more susceptible to drought and mineral deficiencies. And soils with larger proportions of clay are not desirable for most plants because they provide poor drainage and often do not provide enough oxygen. This is why they say that Mississippi mud or clay is some of the worst stuff on earth to, of course, be in sometimes. So clay soils are used in agriculture, tend to compact, which reduces the number of soil spaces that can be filled with air and water. Alright. So clay particles are negatively charged and they attract reversibly and they bind positively charged mineral cations such as potassium and magnesium. The organic matter of soil. So with that organic matter of soil, it's composed of litter, droppings, I did say droppings, decomposing remains of plants and animals and microorganisms. So they're decomposed by bacteria and fungi, which release essential mineral ions into the soil. This increases the soil's water holding capacity. So humus, the particularly decayed organic portion of the soil, binds mineral ions and holds water. Pore spaces occupy roughly 50% of the soil's volume and are filled with air and water. Water is usually held in smaller pores and air in those larger pores. So oxygen is required by soil organisms for aerobic respiration, and nitrogen is used by nitrogen-fixing bacteria, and carbon dioxide is involved in soil weathering. So as a result of respiration, there is less oxygen and more carbon dioxide in the soil than in the atmosphere. This is why it's so important to have soil and not just say, oh, that's dirt. So the soil water contains low concentrations of dissolved minerals that enter into the roots and plants along with water. So water is not bound to the soil particles or absorbed by roots, percolates down to the soil, leaching dissolved minerals. So I say again, water that's not bound to those particles or absorbed by roots, it percolates down through the soil and then leaching dissolved minerals. So some substances completely leach out of the soil, and other leach materials may be deposited in the lower layers of the soil, alluviation. So fertile agricultural soil contains millions of microorganisms, as well as microscopic nematodes and other worms. Other organisms also colonize the soil ecosystem, including earthworms, insects, snakes, groundhogs, and moles. Earthworms, I tell you class, I repeat, earthworms are some of the most important organisms living in soil. And their castings bring minerals up to the surface, and their tunnels aerate the soil. So when I saw earthworms in my soil here at the house, I was utterly amazed and elated. And then, of course, I think it was yesterday, I saw an earthworm that died on the gutter, and I said, oh my gosh, I wish he would have made it, or I wish she would have made it. I should have put him in the garden. So that is it, and it is that. I cannot give you everything. What I can do is say, class, please make sure that you all are preparing for class well. And as I do say that, there was one last thing to mention before I go away from figure 3615. Soil pH. Sometimes it's asked, what type of soil should I have? Well, the type of soil you should have is soil with a pH that ranges from about 6 to 7. And air pollution, such as sulfuric acid and nitric acids, they're produced by us being humans. They affect and cause acid rain, sleet, snow, or fog. And it can seriously decrease soil pH. It has now been a large contributor to forest decline, and there is a strong correlation that has been demonstrated that, of course, between forest damage and soil chemistry altered by acid precipitation. It is real. There are 10 macronutrients required by plants in large quantities. Before you, carbon, hydrogen, oxygen, nitrogen, potassium, calcium, magnesium, phosphorus, sulfur, and silicon. 9 micronutrients are needed in trace amounts for normal plant growth and development. I will go through all of these, but of course there is chloride, iron, and sodium, and I'll leave it at that, but these are micronutrients needed in trace amounts. Not that they're not as important. So I guess what I'll do now, class, is get to, there are 15 remaining essential elements that are obtained from soil as dissolved mineral ions. So I say potassium because it's there for the turgidity of cells and the opening of stomata. Plant cells must remain turgid. If they're not turgid, they're not normal. And of course, they may very well be flaccid. Calcium ions are there for membrane permeability as well as being secondary messengers. Magnesium is part of the chlorophyll molecule. You must have it with those rings for the processes of photosynthesis. And finally, sulfur is found in certain amino acids and vitamins. With that, class, prepare for your final exam. Somebody may ask, how? Let's find out how. On the final exam class, I call it homework, you should be able to describe the process of double fertilization and its importance to plants. You all should be able to describe invasive species and their effects. I would like to know what is an invasive species and what are its effects. To earn full credit, you must use 2 named examples in your description with their effects. I would like to know what was proposed by the intermediate disturbance hypothesis. Go back. You will also provide the differences between the 4 major groups of plants, including the mode of reproduction, spores or seeds, etc. The presence or absence of a vascular system. Be very specific. The dominant generation. Is it a gametophyte? Is it a sporophyte? And at least 2 examples. And note, class, those are the least most requirements. And in saying such, if you just do that, it is not enough. Thanks a lot, humans who damage the soil and erode soil and cause things such as we see here. with this, we'll get into the last chapter for the test. We're not quite there yet, but we will get there pretty soon. So as we do get there, I'll ask you about the way in which reproduction happens in flowering plants. So table 37.1, the floral characteristics associated with various animal pollinators will be on your test. It will be there in, I would say, its entirety. However, there will be blanks. You will fill in those blanks. So please study it and study it well. Along with that, study what is called the alternation of generations for angiosperms. You will have to annotate that diagram. All parts and pieces, the numbers and letters. Next up, it says you all will trace a drop of water through a plant, beginning with the soil and ending with the atmosphere. Please be very specific using information taught to you all from both the 35th and 36th chapters. You'll also trace the flow of sugar from source to sink. It requires energy, you all. Please don't forget it. You all will describe the 8 defining characteristics of animals. Yes, I did say all 8 of those things. You all will describe competition. In addition, you all will contrast what is intraspecific competition and interspecific competition using named examples. And please do not ever forget the thing that is known as competitive exclusion. I will ask about that thing there too. From here, I was going to add to the homework. Where was it? I was going to add to the homework. I think that was it. I thought about adding more. but I don't know that I will. Just in case I do add it, make sure you all are able to describe at least 2 ecological effects of wildfires, and describe 2 pros and cons of fire suppression. And finally, how do controlled burns help the environment and or species in the environment? And if you have not yet figured out, class, I'm referring to plant species. It is not that I don't care about animals, but of course I'm referring here to plant species. Thank you all for listening. This has been the 36th chapter. We have 1 chapter left, the 37th chapter. It is called Reproduction in Flowering Plants. Take care of yourselves, class, and thank you all for listening.